Hey students, so for this lab, you are going to be working on a new pandemic. It's called an influenza pandemic. And you have this safety sheet with different instructions, and as usual, be careful, don't put things in your eyes, have general safety with the entire kit. So please read this over and make sure you have all the necessary pieces, and contact Science Takeout if you are missing pieces. On the first page, you are going to read over this background information because once again you're studying a pandemic. To answer the first few questions, you're going to be using this colored graphic paper where it separates out the seasonal flu and the pandemic flu and finding out the differences between the two. So make sure that you read over that and you understand well. Don't gloss over it. Make sure you really understand the difference between the two. This is something that every year, as there are different illnesses that crop up, that people get really confused over pandemics versus seasonal things. Then you're going to move down and you're going to find 4 pieces of information that you will need to underline in the text box that is different between the two. I want to point out that I don't know why I skipped over this, I shouldn't have, but for my class, if you include some pictures, and that can also be including like pictures of your models as you go through, I will give you extra credit for doing that. And that just helps me verify that you really did do the lab. So if you put fun pictures of you, or you take, a selfie with the lab, whatever, and then have some pictures of your setup as you go, I would appreciate that enough to give you some extra credit on this assignment. So then you're going to write additional information that you learned from this and the previous reading. So this first part is very background. Going on to the second part, part 2, again, don't miss this reading at the top. Make sure you do it. And then you're going to actually do some of the chemical experimentation. So you have a vial here of a patient virus sample. You have a clean pipette. Make sure you keep that clean. Remember, you should try to never touch the pipette to the sides of anything. You're going to line up your well just like it says here with the tab over to this right side. It's very clear in the directions with that. The tab where you can hold on to it here is on the right hand side. That's critical because all of these wells have different chemicals inside of them that will change different colors when you add in this patient sample. Again, because this pipette is going to be used for the patient sample, you don't want to touch it to the insides of the sample because then you can cross-contaminate chemicals. So what you'll want to do is drop from up above so you don't cross-contaminate. So there is some important reading steps here that I'll need you to look over. And you're going to draw X's on the wells up here on your paper version for the ones that changed to a pink color. So let me see if I can move the camera down a little bit so you can see that direction, the ones that turned pink. Okay, then you're going to do some analysis on that. You're done with the testing portion, so you can put that to the side, and you're going to move on to part 3. In part 3, the important pieces are that you are going to be really labeling your cups. This is somewhat similar to what you would expect a lab scientist to do. So for example, the labeling portion and keeping samples labeled properly for patients is really important. You are going to follow these directions. So this first one for like the human seasonal virus, it says human seasonal virus. You're going to use that. You're going to put in 6 blue fuzzy stems, a blue sparkle, and a blue twist tie in the cup. So you're going to be doing exactly what it says and adding all of those things. I didn't necessarily do it correctly here because I don't want to give it away, but you can you can read those and do that. Then it also has some sticker strips that you will be applying to the outside of the cup. You're going to be doing that for humans. You're going to be doing that for the pig, the swine, and the bird, the avian. And you're going to build a model here of the illness. So we're going to move on to page 4. You're going to complete that setup. For this part 3, you will actually have this awesome paper here to show you where you will end up putting the trays and the different cups with your different model things in them to create your models. So this, as long as you are, and I love pictures of this, for example, but as long as you're following the directions here, it'll have pictures and tell you exactly what to do. Don't skip different parts, and you should be fine making your diagram. So this page, page 5, continues with that, where you will make you'll be twist tying different things to each other to make different models. And then on page 6, you'll have to start answering questions about those models and the color coding. And here, number 12, 13, is some are directions that you should do. Some of them are things that you'll need to answer. 15, it continues. This is on page 7, but number 15. So it'll ask you to support your answers. When it says support your answers with observations from the models, I'd love to actually see your model laid out right here on top of it, and then your answer here, so I can see what you saw. That would really help me, and that's where it is worth some extra credit for me, because it's a little extra effort for you. Okay, going on to part 4. This is where you start analyzing some data. So you're going to read this information here, and you are going to look at the seasonal flu. You're going to use a model chart, which, well, first, I'm sorry, you're going to actually draw an antibody. That is from your reading here, that you'll have this big blank space for your drawing and answer a question about that. But then on part 5, you'll look at this graph of the number of cases of the flu. I want to explain this graph a little bit. These are different weeks. These lines, like week 1, week 2, week 3, week 4, week 5, 6, 7, 8, and how many people caught the flu each week. So week 1, it's like almost exactly between 50 and 100. So that would mean about 25 people had this flu week 1. So, and then it went a little up week 2 and week 3. This becomes really important, how to follow this line. Now, separately, there's some dots on here, and I almost wish they would have color-coded those separately, but I want to explain this. These are dots when the scientists made various discoveries or breakthroughs on this. So the first one is the identification of the new virus. So week 1 or 2 is about when they said, ooh, there's a new virus. Here, from here, so week 2 all the way to here, which is week 9 almost, is when they started to preparing a virus strain for mass production. So they started making copies of the virus, scientists did. About here to over here, the virus is incubated, that means grown and processed. And then about here to here, the virus was chemically inactivated and manufacturing of the vaccine is made. So you need to be able to read that. Additionally, another important part of this graph that you'll have to do is you will actually have to add up how many people got the virus in week 1, how many people had it in week 2, how many had it in week 3, week 4, week 5, and you're going to end up having to add up all of those numbers for later on. So really make sure that you understand that sheet. So towards the end, it starts asking you some good questions about if the flu is so low by a certain time period, And that's when the vaccine finally comes out. Should we still vaccinate? And those questions can be answered if you look at this information here. So please make sure you do your reading. That's it for this lab. If you include your photos, I will happily give you extra credits. So good luck on this pandemic lab. Take care.